Welcome to Brain Ponderings, your window into the world of neuroscience research with conversations with experts in their particular area in neuroscience. Today, it's my great pleasure to welcome Nick Betley. He's up in Philadelphia, where he's an associate professor of biology at University of Pennsylvania. He, as you'll hear today, has an interesting trajectory of his research, which has led to a recent paper in the top journal in neuroscience, Neuron, where, and this is where this podcast will end with that paper. We'll spend quite a bit of time on it, but it's really a very new concept, really, and evidence for it that neurons in a particular region of the brain are critical. And I'm not talking about neurons involved in controlling muscles. Uh, Neurons in a certain region, evolutionarily ancient region of the brain, are critical for endurance, physical endurance with exercise training. So welcome, Nick.
Welcome. Thanks for having me. It's a pleasure to be here, and I look forward to our conversation.
You didn't start out working on the brain and exercise. Can you talk about, well, your background? Where did you grow up? How did you end up in Boston University and then go from there?
Yeah, so I grew up in Rhode Island. My mom was she stayed at home. She was a school teacher. My dad worked as an engineer. They were both neither one of them actually went to four-year college de novo. They ended up getting four-year degrees, but started with associate degrees. But were absolutely convinced you have to go to college. So I mean, I went through high school, attended college, and I think I wanted to be a doctor when I went
Uh,
to Boston University. And I think the biggest change of my life came in around 1999 when put in an application the night before it was due. I wrote the whole application the night before it was due to do a summer research project in a lab. And honestly, looking back at it now, I'm not even sure why the professor it was Jim Deschler, my first mentor in science, who's now at the NSF, why he even engaged me. I came to him Friday at 3 p.m. and was like, "Hey, I want to put in this application. It's due tomorrow at 9 a.m." But we wrote it overnight together, and I was the
oh,
first
really?
undergrad in his lab, and he had a small lab at the time. And March of 1999, I remember as the first experiment I did because I had solution bottles labeled with that date in the lab. So that's my like birth date in doing academic science and academic research. Um, and once I got in the lab, that changed everything. I mean, everything I wanted to do after that changed going forward. And I joined science at a really fortunate time. So 99 was around the time the NIH budget was doubling.
Yeah.
And at that time, I saw this amazing future where I could ask questions about the nature of biology, and it's supported financially and, conceptually by the country and our lawmakers. And I really like it was an exciting time to get involved in science. And every day I woke up just wanting to ask a new question.
We were very fortunate in that regard. I started out my first faculty position was in 89 and then that was during the ensuing years is when
Doubling, yeah.
the budget came up and I say we're very fortunate because, for example, one of thousands and thousands, tens of thousands of people, my son this past January, a year ago about, got his PhD
Uh-huh.
from Hopkins in biochemistry,
Cool.
biology. And it's bad timing though,
Yeah, I know.
because all this funding was withdrawn and it's hard to find positions. So now he's doing AI related stuff, but it's related to biology. It's kind of interesting, but it's for a
Yeah,
big company.
it pushes you in different directions. And I could imagine trying to find an academic lab to do a postdoc now is no small challenge.
No, yeah. Okay, so then you s- you started out. I think I look back on PubMed at your publications. Your first one was on frog eggs, frog
Okay.
oocyte.
Yeah. Yeah. Xenopus oocytes. We were looking at how polarity is set up in the developing oocyte. So What a lot of people don't realize when you look at an egg, an early egg, is that, especially if it's not cut, if there's no pigmentation, is that it looks round and similar. But frog eggs actually, when they become before they are hatched as eggs, they actually have pigmentation on one side and white color on the other side. So you can already see there's a difference between the top and the bottom, right? Half of it looks different. But we were studying what happens even before the pigmentation happens. So different things must get there to provide that
So
pigmentation.
this is before fertilization.
Way before fertilization. This was like the earliest stage oocyte. So we were studying stage one and
Huh.
stage two oocytes. And it turns out that transcripts, RNA molecules, are moved to different parts of the oocyte very early on, and that sets up protein gradients later on during the oocyte maturation, and then obviously during development. So you get, a head in the front of the animal and a tail in the back of the animal.
And then all the sperm does is then set everything in motion.
Just yeah, then cleavage starts
Yeah.
as soon as the DNA from the sperm's transmitted. Yeah.
Yeah. Interesting. Okay, and then from there you went up, I think, up to New York City.
Yeah, yeah. So I ended up moving for graduate school to Columbia University. And it seems like a leap to study neural circuit development after that. But it's still, in the concept of science, it's still development.
Yeah.
And what really attracted me to Tom Jessel's lab at the time, um, it's funny how science works. What attracted me was something the lab did 10 years prior because it was in the publication record, and the lab wasn't even really working on that exact question anymore. But they had looked at some of the molecules that we were looking at that were polarized in oocytes, and I was wondering why they're involved in patterning the central nervous system. And so that's why I engaged his lab at the time. I was like, this would be really interesting to study in terms of patterning, and my project ended up being somewhat related, but not on those molecules. It was really trying to understand how a neuron finds its appropriate synaptic target in a sea of hundreds of possible targets. So how do I talk to you instead of
Yeah.
99 other people?
Yeah. And it's complicated too. But
Yeah, I mean, it's funny because when I started the project, I was like, okay, I'm going to find the molecule on neuron A that talks to the molecule on neuron B, and that's how it's going to work. And that's actually, we never found one molecule that dictates the process completely. We did find molecules that would influence it, but it seems to be that it's a combinatorial code of a lot of different molecules and a lot of other factors that set up way before these molecules are even expressed.
yeah, there has to be a sequence of events that where one event depends on the preceding event and so on.
And when you think about it, then it's amazing that we have functional brains in the end, right? That
Well,
always
it
goes
does
right.
kind of make sense from the standpoint of it's a way to generate complexity,
Yeah.
from over time by these sequential
Get these
interactions.
changes and then also to what it leads to in some ways. I guess that's the same thing as evolution, but we all have these individual differences in how we're wired, and eventually those that were most adaptive were taken up and led to evolution of species. So I
Yeah.
mean it gives another opportunity besides genetics for evolution.
All right, so then now you were working on the mammalian nervous system in your PhD work at Columbia, and then you went to Genelia Research Campus. Can you? I don't think I've had. I may have had someone who went there. I know them best for their development of new imaging methods and new technologies for imaging at high resolution, spatial, temporal, in living animals. Um, it's interesting, Nick. In my postdoc work, I uh, studied regulation of growth cones by glutamate,
Huh. Yeah.
excitatory transmitter. And that was right at the time in the mid 1980s when Roger Chen came out with FIRA2, the first fluorescent
Amazing.
calcium imaging dye. So you can, in individual neurons, and I was just doing this in cultured neurons, you can look at the relative amount of calcium. Actually, you can get pretty precise measurement of the concentration and the distribution in the cell. So I was looking at, you know, how glutamate elevates calcium levels in neurons, how that controls their growth cone. So I've kind of followed the imaging
Mhm.
story. And I think when we get to talking about your work on the brain and endurance, you did some calcium imaging with more sophisticated
Yeah, in vivo.
technology. So what did you do with Genelia? That's in Virginia?
Yeah, that's in Ashburn, Virginia. It was on an old farm. They used to call it Genelia Farm. It was a really an interesting place to do research, where basically doing high risk research was really supported by Howard Hughes Medical Institute. There was the people who developed, there was probably two parts of the campus. There was the tool development part where people were developing, as you said, genetically encoded calcium indicators, sensors, and high-end microscopy. So Eric Betts has won the Nobel Prize for work he did. I guess that just preceded his time at Genelia. But I mean, they were developing super resolution microscopy at the light level. So all of this was going on at this campus. But there were also people who straddled the border between tool development and application of tools. And I joined one of those labs, Scott Sternsen's lab. And at the time, Carl Servoda was there, Nelson Spruceton, Jeff McGee, and there was this energy around really trying to use the emerging tools in neuroscience to answer long-standing questions and apply them to hard questions that hadn't been asked before. And that was what actually drew me into Genelia. So when I was studying spinal cord development, we, figured out neural populations that how neural populations get wired at some level. And we always wanted to know, well, what are these neurons doing? What would happen to behavior if we modified the activity of these neurons? When are these neurons active? And we couldn't answer those questions at the time. So it was a little bit frustrating. We had to infer, and this in many ways is kind of the trend in new science, right? We don't have to infer as much anymore because we get overwhelmed with data. We're actually data rich now, whereas before
Yeah.
people used to, I always say that people who were scientists 40 years ago, they actually had all the right answers, but they just had to do it by like theory and thinking about logically how things, what's connected to what region of the brain and how can that influence behavior based on lesion studies.
Yeah.
And so it's almost embarrassing. Like we almost don't have to think anymore because we can just overwhelm this with data. But on the other hand, it was liberating because we can actually directly assess the question.
Yeah.
And when I went down to Genelia to visit, I was um, walking around Scott's lab with Dennis Adezoy and Yeka Aponte, who were two postdocs that preceded me. And they brought me to this little closet of a room in the back where there were all these wires and lasers shooting everywhere. And the wires eventually made their way into the mouse's brain. So you would connect the little fiber optic to a little ferrule in the top of the brain, and they turned on the laser. And this mouse that was just chilling out went and just started eating as if it had been starved. And I mean, this was kind of like the old LH stimulation experiments of Old and Milner. And this just drew me in. I was like, wait, what are you doing? And they're like, we're activating just 1,000 neurons in the brain. And I'm like, 1,000? And the animal's doing this? And so I mean, I had to know more. That I knew 5 minutes after seeing that I was going to go there and I was good. This was a system that I could do a lot with in the future. And naively, what I thought that day was that this is also a very tractable behavior, right? Feeding, like we can measure how much food the animal eats or how much less they eat over the course of it. We can weigh their body weight, look at their body composition. And I was always daunted by trying to do neuropsychiatric experiments in a mouse. So I liked the fact that this was grounded in homeostatic behavior that had really easy readouts. And as you get into it, you find out that you're actually confounded by all the same things that you're confounded on with neuropsychiatric research in mice. And when you try to ask, how does this feel? When the mouse's neurons become activated, you realize you're fighting the same battle everybody else is. And it makes me think back to some of the older pain researchers who at some point would stimulate their own nerves just to understand what it felt like to have stimulation like this that they do in their experimental models. Up until that point, I couldn't understand how anybody would stimulate their own nerve. And then after seeing the animals do this for 5 years, I'm like, I wonder what this feels like.
Yeah. Yeah. And and those are big. I mean, obviously, hunger and pain are very critical for survival. I mean, being able to
Hmm. To sense them and respond to
to
them. Yeah.
sense them and hunger to get food. And now in our modern environment, though, you can be hungry too much and eat too much.
Yeah.
Right. So what were your experiments there at Janelia? You published a good number of papers.
Yeah, it was a really intense couple of years,
Yeah.
and I got focused on the hunger neurons in the hypothalamus known as the AGRP neurons. So there are 10,000 of these neurons, and the first paper I published kind of took my developmental approach to neuroscience, and these neurons target regions of the forebrain, the hypothalamus, and the hindbrain. And I just looked at the projections of these neurons throughout the brain, right across 10 millimeters of neural tissue. And I was like, does the neuron that goes left also go right? And I was like, there's just no way in my mind that the neuron, and this reflected a naivety in my understanding of neuroscience, right? Because there are these dopamine neurons that project throughout the entire brain. But fortunately, I hadn't read those papers yet.
And serotonin neurons and
Neurons,
norepinephrine.
yeah, all of these neuromodulatory systems have this massive trajectory. But there was no AI for me to ask this question at the time, so I didn't get a report saying like, yes, Nick, there are five populations of neurons that do this. So, you know, I was and PubMed was online. Everything was slowly getting online at that time, but it still wasn't as easy to search back then as it is
Yeah.
today. So I had this naivety that led me to believe, my own instinct that the neuron that turns left isn't going to turn right. So we started looking at the connectivity and the function of these neurons. And it turns out that in this case, there were dedicated populations of neurons that went to each of the target regions in the brain. So they don't form these massive collateralizations like the neuromodulatory systems. So that led to the question, well, why do you have all of these separate populations? There must be something that the neurons that project to, say, the extended amygdala do differently from the neurons that project all the way to the hindbrain and the parabrachial nucleus. So because they were separate neurons, we were able to optogenetically stimulate them by using one of the tools that was developed in the Dizeroth lab, channel rhodopsin. So you put this protein in that's basically turned on by the switch of a light. And you put light over the axons instead of over the cell bodies, and you can just activate the neurons that project to that one region of the brain. And we found that a bunch of them are important for feeding, but there were three or four axon projection populations that had no effect on the animal feeding.
Okay.
And we were like, it was always a question. In fact, when we went to publish the paper, I was obsessed with trying to find a function for them because I was like, nobody's going to like this paper if you don't know what the heck these neurons are doing. But as it turns out, that was actually the only paper I've had published since my very first paper that went through review without a revision. The reviewers actually accepted it without revision.
I don't know
Yeah,
if I don't think I ever had that happen.
yeah, it doesn't happen anymore.
Yeah.
I mean, I was shocked. In fact, the editor was so surprised that he wrote us an email, and we didn't know what to do with this email. The editor was like, "Here's your reports of the review. What do you think we should do next?"
Wow. What do you mean? Send it to the publisher.
Well, yeah, no, I mean, eventually that's what we said, but we looked at each other nervous. We're like, "Is this a trick? Like, what do we do?" Yeah, but I think the editor just never seen a report like that in recent times, so he just was confounded as much as we were. Anyway, so
So, so there's a subpopulation of these 10,000 neurons, AGRP
yeah,
neurons, that if you activate them using this new optogenetic method, the animal doesn't exhibit any hunger behavior. Is that right?
yeah, yeah. So
And then, and as you said, then what's the question? What does the animal experience anything when you
yeah,
stimulate?
yeah, and it took another 7 to 10 years for me to come to some answers on that, and we still don't know what all of them do. But we've in more recent times in my lab have come to the some further understanding of what the AGRP projection to the parabrachial nucleus does. But what's really, interesting in the system is there are, say, 3,000 neurons that project to the paraventricular nucleus of the hypothalamus. You activate those neurons, the animal eats.
Okay.
Other labs in the field have shown that if you activate those neurons, you actually get the elevation in cort in the blood. So the stress, when you're hungry, you actually release cort into your circulatory system, which may enable you to go out and seek food. It's like a stress signal. Um, the There's about 1,000 neurons that go to the bed nucleus of the stria terminalis, which is a region of the extended amygdala. And if you activate those, you also eat food. Uh, but there's about 500 neurons that project all the way to the back of the brain, the parabrachial nucleus. And if you activate those neurons, you do not eat food if you're just sitting there not hungry. And those neurons actually have two functions. So while I was doing this work, Richard Palmetter's lab was studying these neurons, and he showed that those neurons are important for suppressing what he called visceral malaise, what you and me would call nausea,
Hmm.
uh, to eat. So, if your stomach's upset, that or this is actually clinically important for chemotherapy. So the neurons in the parabrachial nucleus are responding to chemotherapy, and that's what causes nausea. Same region of the brain actually becomes activated when you're having motion sickness.
Hmm.
And these hunger neurons release. He showed that the release of GABA is important for them to suppress the visceral malaise in these parabrachial neurons called CGRP neurons. And work subsequently in my lab, um, looking at the intersection of okay, so I mean this would be a gap, but work in my lab actually showed those neurons having a completely different function in the parabrachial nucleus in suppressing pain, and it does it not through the release of the neurotransmitter GABA, but the neuropeptide NPY. So it's a really cool concept that neurons that go to the same region of the brain can have two functions based on the different uh, things released in their synaptic vesicles.
Yeah. But in a way, yeah. So nausea is kind of a noxious thing, and pain
Yeah.
is a noxious thing. So in that more general sense,
Yeah, in a more general sense, is a conserved function.
yeah. Oh, that's very interesting. So if if a person is hungry, they're perceived like if someone had chronic pain and they're hungry, their pain level would go down.
So this is the like therapeutic
Yeah,
question.
I'm
That
jumping
would be,
ahead, but
yeah, no, but I mean, this is a brilliant. I mean, this
yeah,
is what we think about all the time. I think an important caveat to consider to these experiments is that the level of hunger that you need. So when we see this, we've food deprived the mice for 18 to 24 hours, and that's like food depriving a human for 6 or 7 days. So it's a very uncommon level of food deprivation.
yeah,
When we publish these studies, we actually get anecdotal reports from people who, talk about when they were hospitalized and didn't eat for 5 days and how their chronic pain was somewhat alleviated. So it gives this, almost case report type validity to this. But yeah, it's not a therapeutic, hunger alone would not be a therapeutic um, prescription because most people, I mean, you can't survive if you're only eating once every
yeah.
7 days.
We did a lot of work on fasting, intermittent fasting,
Yeah.
and but I haven't seen like a clinical trials where they look at, whatever
Pain,
pain,
yeah.
pain during fasting. It'd be interesting.
I mean, they should. And I think the other interesting angle to this is, you know, people who intermittent fast is as a population, are they less likely to experience chronic pain? So is there a build-up effect of doing this for years?
That one thing that happens during fasting is you switch from glucose to ketones, and one of those ketones, beta hydroxybutyrate, it has signaling functions. We found it enhances GABAergic tone, at least in the hippocampus. And yeah, it'd be interesting to see
Yeah.
what that ketone does.
in the parabrachial
Yeah,
nucleus.
yeah.
Yeah. No, I mean, this is actually phenomenally interesting work that you've been a part of, and it's something we've only started to think about in terms of how the brain's metabolism, or the metabolism of the individual cells, and the metabolites that get produced in different states of hunger and fasting change the biochemistry of the brain, right? All of these metabolites are like allosteric modulators of receptors, or direct agonists and antagonists of receptors. And so the entire chemistry of the brain and the body, which we've gone on to begin to be fascinated by in the lab now, is different in all of these different states, hunger versus satiety. And now we look at this in terms of sedentary individuals versus exercised individuals, which I almost look at as a state of being itself.
And okay, I guess we can jump to the exercise paper. But there's, unless you want to go more, I mean, you did some more related to the anti-inflammatory effects of
Yeah.
hunger. Um, but the way you do the hunger, they're deprived of food for 18 hours, and but you show these particular neurons in the hypothalamus are critical for the anti-inflammatory effect.
Yeah, so
Yeah.
with the pain work, we got interested to see if there's um, an effect also. So we had been studying longer term modalities of pain, and one of the main assays we used was a what they call subacute inflammatory pain. So the inflammation and the pain, or the pain behavior the animal exhibits associated with it, lasts for about an hour and a half. We've also done models where the inflammatory pain and the behavior associated with that lasts for a couple of days to a week. And we were just wondering, like, when you get an injury, right, your hand gets puffy, and like, is how much does that contribute to the pain? And are the AGRP neurons actually shutting off the pain by shutting off the inflammation? So we were studying that, and it turns out, so I had told you that the hunger neurons that project to the parabrachial nucleus suppress the pain. It turns out that the neurons that project to the paraventricular hypothalamus reduce the inflammation, but they do this without reducing any of the behavioral signs of pain. And so the mouse is still attending to its wound, the mouse is still shaking its limb. Um,
Ah,
so it doesn't seem to affect the actual pain that the animal is experiencing, but the inflammation goes down. So
interesting.
yeah, so different subroutines that these neurons encode uh, help alleviate these different aspects of a response to an injury. And I
Interesting.
think, what was most fascinating to me, we recently did the final, no, not the final, the most recent update to the pain paper in the parabrachial nucleus. And you know, what we think is going on in the parabrachial nucleus is that it is a bottleneck in the brain through which pain information is sent from your spinal cord to the brain. And from there, it's distributed to the rest of the brain. But if you can cut it off at this bottleneck, you can reduce the effects or the feelings of pain. And I think, that's where it provides a clinical opportunity to see if, there's a way to, like you were saying, can you upregulate GABAergic tone in the parabrachial nucleus, or peptidergic NPY tone in the parabrachial nucleus, and shut these neurons down?
Yeah. And I had Kevin Tracy. I don't know if you've come
Yeah.
across him. There's a
Yeah.
lot of work on the vagus nerve and inflammation. Are there signals coming through the vagus nerve that get to these neurons in the hypothalamus?
Yeah, so this was kind of one of the first experiments done in my lab. We were still studying hypothalamic hunger neurons, and um, what I had shown in subsequent studies in my postdoc, um, in a collaboration with the guy Shenzhen Zhu, who's in the lab at the time, he was doing calcium imaging at a single cell resolution in the arcuate nucleus of the hypothalamus, and he had this finding that I'll never forget these lab meetings. As soon as he showed the animal food, the neurons would shut off. And this finding was actually repeated by three other labs in the field, all at about the same time. But I remember when we were looking at it, we were like, oh, it has to be like the animal's moving and the fibers jiggling or something. Like, nobody really could believe that looking at food shuts off your hunger neurons. And so
Yeah.
Right? Yeah, it doesn't
Yeah,
make
yeah.
sense. Like when I show this, I can show this to high school students, and I'm like, what doesn't make sense about this? And eventually they're like, well, you haven't even ate the food yet. And I'm like, yeah, like this is a problem. And so, how I view this is your brain is largely anticipatory, right? And if you've been at a restaurant waiting for a table, you finally get a table, you sit down, you take your order, it's really busy. You start getting this anxiety, like, am I ever going to eat? And you're looking around, right? And then you see a waiter or waitress come out with a plate of food, and immediately you're relieved, right? You're like, oh, I'm going to get to eat. Now, what happens when the wait person walks by your table to the next table? It all comes back. So that's actually what we found was if you don't actually eat the food within 300 seconds, the activity of these neurons comes back. And if you give the animal celery, in our case, we gave it nutrient-free, calorie-free Jello. So the animal will eat this because it's sweet. They'll eat a lot of it. The neuron activity goes down for about 200, 300 seconds. And even while they are still eating this calorie-free Jello, the activity of these AGRP neurons comes back within 300 seconds.
But if they eat calorie containing food, it
Stay
stays.
down. Right. So that told me that there has to be some signal from the body to the brain that's keeping these neurons off and suppressing their activity for a long period of time. And, so then the hunt was on for how are these neurons signaled from the body? And so at the time, Amber Alhadeff was in the lab and she wanted to do an experiment where we gastrically infused nutrients directly into the gut of the animal while looking at the neural activity in the brain.
Mhm.
And I remember being a little bit afraid of doing this because the lab wasn't, I had just become a neuroscientist. I wasn't a physiologist. We didn't know how to do peripheral surgery, but people in the field, Gary Schwartz especially, was super helpful. He brought us up to the lab, showed us how to implant these catheters. And, within 2 weeks, we were doing experiments where we were infusing food directly into the stomach. And this was probably the most shocking experiment because within 30 seconds of the beginning of infusing food into the stomach, you could see the AGRP neurons shut off. So that meant that it's not, glucose getting into the blood and diffusing to the brain over the course of minutes to tens of minutes. This was happening very, very quickly. And so we reasoned it had to be a gut-brain pathway. And it turns out that if you lesion the vagus nerve, you completely get rid of the ability for the brain to respond to gastrically infused nutrients. So the vagus nerve is of critical importance to this.
So is it like physical distension of the?
No, so we did a control for that. We put
Okay.
methylcellulose into the stomach to distend it
Uh,
as much as possible. So you do that instead of water because you need, if it's water, it'll pass through the stomach very quickly. So you basically fill it up with basically celery, the cellulose. And there was no effect on AGRP neuron activity.
I see.
So what turns out that it's dependent on the amount of nutrients you put in. It did not matter whether the nutrients came from fat, sugar, or protein.
Huh.
It's a calorie dependent mechanism. Now, subsequently, we showed that there are different pathways that signal fat versus sugar, and that there's all different molecular mechanisms, right? The stomach's uptaking these molecules differently, and it's signaling the vagus. But at some basic level, if you put enough of calories in, regardless of what they are, they will shut off the AGRP neurons, and they'll do it with about equal efficiency.
Interesting. Okay, let's get to the exercise, which is
Yeah.
actually the reason I contacted you because I was uh
Yeah, because you're
well,
into exercise.
yeah, well, no, that isn't the reason I contacted you. You told me about your neurons. It hadn't come out. I invited you right when it was going to come out the next
Yeah.
week. And so I think this is going to raise a lot of interest and questions. There's one big question. There's always new questions, but okay. So everyone knows that if you exercise regularly, you get in better shape. You can improve your performance over time. I used to run a lot,
Hmm.
and it takes a while to get in shape, several weeks to a month before you really start to notice it. I think that's why a lot of people they may say, I want to start exercising, and then they just do it for a few days and maybe don't feel so good because they're not in shape. Um, and I think the conventional wisdom is okay, your muscles get stronger, better endurance because the muscle cells themselves are being stressed in a good way, and they respond to that stress over time with repeated cycles of exercise, rest, eating, with they get physically bigger, more protein components in the contractile apparatus, more mitochondria so that they can generate more energy. And so at least my take on it is that was kind of the general thinking about how you get how what endurance is and how it gets better with repeated exercise. So
Yeah.
so go ahead and talk about your paper.
Yeah, and I mean that was our general impression when we started the paper, when we started the study. So we got into exercise basically out of curiosity, that was driven by the fact that a researcher in my department had a treadmill sitting in the lab, a rodent treadmill sitting in the lab. And we always wanted to know what the heck goes on with like hunger neurons when you exercise, right? Because
Yeah,
when I exercise, I'm not really hungry right after.
yeah,
Then later I get really hungry, but like for the half an hour after, I don't want to eat.
yeah.
And it wasn't our main focus, so we never used the treadmill, but it was always sitting there. And I got into this field because that was always in the back of my head. And two different people I had run into 5 years ago were studying exercise, and one of them was Kevin Williams at UT Southwestern, and he was studying it in AGRP neurons using electrophysiology. And I told him, we have these mice, we have a treadmill. I'm happy to run them for you. We just want to know the answer. You can use the data. And so we did that great collaboration with him that's led to many more collaborations. And at the same time, a guy who was in my diabetes research center in the microbiology department at the time, Christoph Theiss, was sitting behind me at a seminar talking to his technician about the dopamine changes in the brain after exercise. And it was the same conversation. I look back at him, I'm like, what are you doing? And I'm like, we have these mice where we can sense dopamine in the brain in real time. We can run them and see what's going on. So we kind of unwittingly got pulled into this field, and what we started to discover was so captivating, it kind of sucked us in. In collaboration with Joel Elmquist, we participated in a program project grant looking at the effect of the hypothalamus, or how the hypothalamus is involved in exercise. And, largely going into it, we thought that what we were going to see is very similar to what people had expected, that the changes in the brain are a consequence of exercise, right? You
Yeah,
exercise,
improve mood
yeah,
and actually mental clarity,
cognition,
mental
mental, yeah.
clarity. Yeah,
Right. Neurogenesis occurs. There's more synapse. All of these good things. Depression is lower if you exercise. Anxiety is lower. So there's all of these positive effects, and we were trying to figure out how the hypothalamus was involved in that.
I see.
Um, and we started recording the activity of these VMH neurons called SF1 neurons. We were inspired to do this by work done by Tepe Fujikawa in Joel's lab because he had knocked out the transcription factor SF1 from those neurons before we started, and the mice weren't as good at improving endurance. They improved much slower when you knocked out just this one protein. So it suggested something about these neurons is important for the development of endurance. And so we were just using our tricks of using calcium imaging to monitor the neural activity dynamics in these neurons. And we would do this before, during, and after exercise. And we literally, it's actually kind of funny to watch the videos. We'd put the mouse on a treadmill, they'd get acclimated to it for 10 minutes, and we'd start the treadmill and they'd run. And they don't run like us. They'll like do a sprint and then they'll go back to the back
Yeah,
of the treadmill, do another sprint. But they'll do this for 2, 3 kilometers. I mean, they're amazing. They
yeah.
can go so far. So we do this, and we noticed that at the end of exercise, right when they hit exhaustion, when they can't go anymore, the activity of these neurons spiked. And the activity stayed on for 10s, 15, 20 minutes. It's a little hard to put a precise number on it because the technique we're using, while it's awesome and lets you do, imaging in a awake behaving animal, you don't get temporal and the precise nature of you can't compare action potentials before and after event. It's just more relative levels of calcium. So, but anyway, we could see even the calcium was elevated for at least 20 minutes after exercise. And so we were like, okay, well, that's really interesting. What happens if we um, train them for a week? When we did that, we noticed that the calcium dynamics in the neurons or the neural activity after exercise was doubled, and it lasted even longer. And we were like, okay, so not only is exercise changing and remodeling your muscles and making them stronger, but it's changing and remodeling your brain and making these circuits in your brain stronger. And so we did a pretty deep mechanistic dive on these neurons. So we collaborated again with Kevin Williams at UT Southwestern, and he was able to show by comparing brains of exercised and sedentary mice, taking out their brains and recording from the VMHSF1 neurons, that there's twice as many action potentials in um, these slices on SF1 neurons of animals that were exercised versus sedentary. We also did some anatomy with Eric Bloss at the Jackson Labs, who did array tomography to look at dendritic spines on SF1 neurons. And so he sees a doubling of what
Wow.
you could interpret as the excitatory input to these neurons on the spines. So there's more synapses forming on these neurons, there's more activity in these neurons, and after exercise, it allows there to be a bigger burst of activity. So what are these neurons doing? We had to know. So we did an experiment where we silenced these neurons after exercise. So the animal takes its run, and we silence the neurons as soon as they get tired. And we do this for a week of training. And then we measure their endurance. So we measured their endurance before, and then we measured it a week later. And their ability to gain endurance was greatly compromised. They could only basically do 30% more of more that they could only improve about 30% of what a normal animal could improve. And we looked for why. So some of the things that are different. Normally after exercise, there's this massive, you talked about the remodeling in the muscle, and I think that's underlined by a massive transcriptional change. So
Yeah.
there are master regulators of transcription that change in the muscle, like PGC1 alpha, but they lead to like thousands of changes.
Yeah.
So like 1,000 genes are increased in expression, 1,000 genes are decreased in expression. When we inhibit these neurons, there's like one change. So, you know, 99.9% of the molecular changes in the muscle don't happen.
Wow.
The other thing that doesn't happen, and this was a surprising observation to us, when you exercise, at the end of exercise, there's an increase in glucose in your blood right after. And that actually makes sense from this SF1 neuron perspective because Michael Schwartz had shown years earlier that if you activate SF1 neurons, you increase blood glucose. And so we inhibited these neurons after exercise, and you don't get that increase in blood glucose,
Huh.
presumably coming from the liver. So the fuel that you need to repair your muscles, or I mean, I'm projecting this. We don't know that it's the glucose that's involved, but obviously your muscles torn down. You need fuel to the mitochondria now are very hungry to create ATP to repair the muscles that have been stressed, and they don't have that ready source that gets pushed out into your bloodstream after exercise.
What about the mTOR pathway, right, which is critical for growth?
Yeah, I mean, all of these, every single pathway that that is normally upregulated was just not. And we see this, we're actually looking at this more comprehensively now. So the paper was um, a really interesting observation that we felt we had to share with everybody, and we haven't done every single possible perturbation of these experiments yet, which we normally actually hold them a little longer and do all of this, but it got to the point where we was like, other people need to work on this because we don't have enough of people or enough money to look at all of the different angles to this. So it was time to
Yeah,
share with the
because
world.
there's still kind of a black box between those neurons and the muscle.
Yeah. Yeah.
And, how are they communicating?
Yeah, so there's two huge black boxes in my mind. That is one of them. So what is the muscle? What are the neurons doing that communicate with the muscle? And the second black box to me is how do those neurons get turned on after exercise? How do they know you exercised?
Yeah.
And so both of them are important questions. I think, both of them can be useful therapeutically. Can we, and I think about, obviously after the paper came out, people who are weightlifters and athletes got really excited thinking about like, can they do brain doping? Can we turn this region of the brain on after we exercise and get stronger? But I think about it therapeutically more in terms of geriatrics, stroke
Yeah,
recovery, sarcopenia. And this is going to be a big deal with GLP-1 users, GLP-1 agonist
yeah,
users,
yeah.
especially those who cycle through rounds of GLP-1, because each time you go through a round, you lose the body fat, but you also lose muscle. But when you go off of it, unless you exercise, you preferentially put on body fat.
So
So over,
GLP-1 inhibits appetite,
yeah.
and it's acting on which neurons in the hypothalamus is it acting?
So GLP-1 is active. So the current, yeah, this is a great question.
Okay,
People are still studying this. GLP-1 seems to be mediating a lot of its weight loss effects through neurons in the hindbrain.
okay,
So satiation, it activates the satiation center of the brain. It also activates this center of the brain that's involved in nausea. And you can think about that like in terms of you had a big Thanksgiving meal, right? You overate, you're stuffed, right? That it doesn't feel good at some point. And so that's what GLP-1 is mimicking. But it's, therapeutically, it's amazingly effective at weight loss, right? People who go on it lose 15 to 20% of body weight.
yeah.
So,
But they can
if you're
also they're also losing muscle mass.
Well, so that's where this becomes
Yeah,
that's where the rubber hits the road. One of the biggest predictors of morbidity in old age is lack of muscle mass.
yeah.
Like the clinical test, and I'm learning this with my parents, is can you sit on the floor and stand up without using your hands? Right? That is like the number one right? So if you
I don't
fall,
know if I can do that.
I'm sure I bet you can. You'd be sure, yeah. I mean, you don't have to do it. You're thinking like a yoga
Oh, you
pose.
can go to
You
your
just
knees first.
absolutely. Yeah, yeah,
Oh,
yeah.
okay.
Clinically, yeah. You just have to be able to get up.
Okay.
Yeah. Yeah. So I mean, I think that answering those two black boxes is important. And the reason they're important is because of the very last figure we put in the paper was kind of, it was kind of one of these cute experiments you do to see if it's going to work. And so, um, Ryan Post, who was the postdoc in the lab working on it at the time, I said, just activate these neurons after they exercise for like an hour. Hit them with a ton of this activation. Train them every day and hit them with the activation, see what happens. And you were talking about how people improve. Mice do the same thing. So if they can run a kilometer at baseline without training, by week two, they run 1.5 kilometers. By week three, they're running 2 kilometers, and then they plateau. They
Yeah,
plateau at 2 kilometers and they can't get better
yeah,
unless you change the training. We don't change the training paradigm. If we add in this stimulation, by week three, when your control guys are running 2 kilometers, these guys are running 3.5 kilometers. So it improves at the same training paradigm. It improves your ability to show improvements in endurance, and presumably that's reflected in muscle.
now there's a it seems like there's a psychological component to endurance too, that how much distress can one tolerate,
Yeah.
without stopping.
Well, I mean, so this kind of feeds into that thought, right? Like how much of this is really in our brains?
Yeah,
Right. And people talk about this exercise. David Goggins talks about this, right? Like when you're tired, you've only hit 10% of your capacity. Like
yeah,
you just have to work through it, right? And I think about it, and maybe in a more sympathetic way to myself, but I've never been a profoundly good athlete. I can, with a lot of training, I can get to, the top 10% of my age group, but I can never get above that.
yeah.
And I'm like, well, maybe my SF1 neurons just aren't wired properly to be able to. Maybe I just don't have that capacity. I have a few less SF1 neurons, right?
Okay, so you'll be the first guinea pig for the experiment, stimulating the
My
SF1
SF1 neurons.
neurons. Yeah.
Yeah, I mean, we actively are looking to see if, obviously it's the brain and it's complex, and, what else are you doing when you stimulate these neurons? How does that make you
Uh,
feel? There's a lot of potential ways in which this, it's the story I'm telling is the just so stories, but we're a long way from being able to do this in a human. But we're actively interested in seeing if, what are the pathways to activate this and how can we do that?
what's this transcription factor SF1? Is it widely present in neurons all over the place, or it's
No. No, it's so
And are there any gene variants or mutations that have showed up where in humans where there's a
that
some clinical phenotype consequence?
That's an interesting question. It would also be interesting to look at the protein in athletes versus
Oh,
non-athletes,
yeah.
right? If it's just a gating of the transcription factors, right? If the subtle things that happen are the function of, so it's a transcription factor, it's a nuclear, it's an orphan transcription factor. Um, it is not just expressed in SF1 neurons, but it's not ubiquitous in the brain.
Okay.
And in development, it's important for the proper development of the hypothalamus for the VMH. So if you get rid of the transcription factor, the VMH, the identity of the VMH neurons gets messed up, and it's really hard to compare apples to apples. Like, how does that become? What are the? You can't really draw a line between
Yeah.
normal VMH and SF1. Like, this is the neuron that's been changed anymore because you don't have that same identity. So.
And then what are signals upstream of SF1 that activate the transcription factor?
Oh, the molecular cascades. Yeah, we
Yeah.
haven't looked at the molecular cascades. I think I'm sure the developmental people in the hypothalamus have actually looked at the
Yeah.
development of the transcription factor. We've been so hyper focused on the development of the nervous
Yeah.
system
Yeah.
and where are these neurons projecting, who are their
Yeah.
inputs.
So
So
far, the SF1 is just a marker that you can identify.
yeah, but you're right in that the previous study that it motivated us showed that the decrease in the levels of SF1 was important for endurance. So, I mean, one of the things we're
Yeah.
proposing to do is to look at modulating levels of that protein and record from the neurons and see if there's a link, right? So do the modulation of the protein at the transcript level, but ultimately modulate the amount of protein in the adult. So after the brain's developed, let's upregulate or downregulate the protein at some
Yeah.
level and see how it affects endurance and the brain function.
Interesting. And what have you been contacted by the press much yet?
This story was the most contact I've ever had.
Yeah.
Um, I didn't really know when you submit these things and finally get through a review, you're so beat up that you're not really sure, what the impact's going to be. But this was the most I've been contacted. So I think CBS News did something on their radio program. National Geographic has covered it already.
Yeah.
Um,
Interesting. Good.
there's a general interest in this because, people want to know, I don't know, people want to know what they can do to be healthier. There's a
Yeah.
big interest, I think, broadly in that. And
Yeah.
and I think something that goes along with that is what's the shortcut I can take to get there?
Well, so far there isn't. So,
Yeah.
and also maybe those particular neurons are important for endurance, but yeah, I don't know. Does that mean, okay, there's a lot that goes, physical exercise has a lot of effects on health, right? Improved glucose regulation, improved cardiovascular risk profile, actually anti-inflammatory effect, and all these different things. So I think athletes would be the most interested in getting a head on that. I'm not sure that any manipulation of that's going to be approved by the anti-doping agency. It will be a different kind of doping, I guess. But
Yeah. It's a
yeah,
wild thought. But you're actually tapping into there one of the third important question. Um, you know, we've shown that these neurons are important for endurance and the muscle remodeling, but all of these other effects, the anti-inflammatory effects, the cardiovascular effects, the cognitive effects, is this neuron population a bottleneck for all of the beneficial effects of exercise, or is that distributed in the brain? There might be other populations that are controlling the cognitive effects
yeah,
that, is the upstream regulator. And we haven't done enough of studies to even have insight into that. But I would argue that, if I had to place a bet, I would say that there's multiple pathways and multiple different bottlenecks. And the only reason I say this is because people have been trying to develop an exercise mimetic for 40 years.
Yeah.
right? And there are different things out there that have different levels of efficacy, but none of them are like exercise in a, there is no exercise in a pill today,
No.
right? And that's probably because you have to activate 20 pathways going up.
So if one were to take a isolated muscle,
Yeah.
just remove the muscle, living muscle, and then stimulate it, can you increase the muscle's endurance just by
Well,
stimulating it? And is the act is if that if say that was true, then is the activation of those SF1 neurons in the hypothalamus um, somehow interrupting uh, how would that work? Interrupting a signal, another signal, or
yeah, so okay.
that inhibits endurance? I don't know.
So I mean this is a good question. So here's one of the ways in which I think this is important to think about this. Let's isolate a muscle, but we can do it in your body. Say I only allow you to do curls with your right arm,
Yeah,
right? So your right bicep gets ripped up every day. If you do enough of them, I'm sure your SF1 neurons get activated, right? When your SF1 neurons get activated, why does your left bicep not get bigger? Right? So there's
yeah,
some coincident, yeah, there's some coincident thing that occurs at the level of the
okay.
muscle and a signal that the SF1 neurons lets out. And figuring that out's another great puzzle.
Yeah, there could be, yeah, it's complicated.
Yeah.
But so you have to have both. You have to have both SF1 neurons activated and the exercise. And
Yeah, we actually tested that in the mice.
yeah,
So at what level it affects you without it, you don't build, so you can't just take a mouse, leave it sedentary, activate the SF1 neurons, and it runs farther. That won't happen.
yeah,
We haven't done enough of testing to see if like I give you a really easy training paradigm, right? So, my mom won't run. I can't even get her to walk on a treadmill at a 15-degree incline, but she'll go for a walk. So what if we can, what if she can get a little bit of SF1 neuron activation after that? Will she want to then walk on a treadmill with an incline, right? Like because you built up and it doesn't hurt, right? And
yeah,
so we haven't. I want to do those experiments in the rodents where we,
yeah,
see like what kind of training protocols can be enhanced by the activity of these neurons.
yeah. Interesting. Okay, Nick, I've taken a lot of your time. I know you're busy, busier than I, because
Oh no, this has been a blast.
yeah. And I enjoyed talking with you, and I look forward to see what comes out of your lab in the future.
Thanks, Mark. It was a pleasure to chat, and I look forward to talking to you again in the future.
All right, bye.
Bye. Thanks.